Hello everyone. In previous videos, we have discussed regarding nitrogen fixation by symbiotic as well as non-symbiotic method by rhizobia, by blue-green algae as well as associative bacteria. Now, in this nitrogen fixation process, the nitrogenase enzyme is the important component or important factor. This enzyme catalyzes the reaction of nitrogen fixation. So let's see the structure and function of nitrogenase enzyme. The nitrogenase is a complete group of enzymes encoded by NEF gene, and it has mainly two components. First, the heteroterameric MOFE protein, and second, the homodimetric Fe protein. The first component, that is the heteroterameric MOFE protein, as shown in the figure, which reduce N2 to NH3, that is atmospheric nitrogen, into the stable form of nitrogen. It is also known as component 1. It consists of two subunits, and it consists of two alpha subunits and two beta subunits. Look at here. This is the MOFE protein, or we can call it as the component 1. Here, this component 1 also contains two iron sulfur cluster known as P clusters located at the interface between alpha and beta subunits, and two MOFE cofactor electrons from the Fe protein enters into MOFE protein at the P cluster, which then transfer electron to FeMO cofactor. Each FeMO cofactor then act as site for N2 fixation. This is the site for N2 fixation in the MOFE protein. Each component 1 is also known as denitrogenase component or protein 1 component, or it is also called as molybdenum ion protein, that is MOFE protein. Now, the second component is homodimetric, as the MOFE protein consists of alpha subunit and beta subunit, that is different types of unit. That's why it is called as a heteroterameric MOFE protein. While in case of Fe protein, they consist of same kind of protein. That's why it is called as a homodimeric Fe protein. It is highly reducing. It has high reducing power. This is the MOFE protein. This structure is called as a MOFE protein. This one. Here it has high reducing power and is responsible to supply electron to the MOFE protein to reduce N2 into the ammonia. The Fe protein is also known as reductase. It has one Fe4S4 cluster of 60 to 64 kilodalton weight. This component 2 or protein 2 reductase Fe protein is called as denitrogenase reductase. Both the enzymes are highly oxygen sensitive, and they are damaged or become inactive due to presence of oxygen and stop the function of nitrogen fixation. So, this enzyme nitrogenase is a metalloenzyme as it contains MO and Fe along with proteins. So, this is also called as MOFE protein. This enzyme is found in phylogenetically diverse group of prokaryotes ranging from aerobic heterotrophs, aerobic and anaerobic phototrophs to strict anaerobes. Actually, there are different types of this enzyme, but they are biochemically similar, and hence the complete family is referred as nitrogenase enzyme. Now, Let's see what are the different functions of nitrogenase enzyme and its components. The main function of nitrogenase enzyme is biological nitrogen fixation. Second, the electron are delivered one at a time from Fe protein involving component, protein association and dissociation, and hydrolysis of Mg ATP, at least 2 for each electron transfer. Third one, the site Fe protein contains site for Mg ATP binding and hydrolysis as shown in this figure. So Fe protein has a site for Mg ATP bonding and hydrolysis. Here, look at here. Now the site for substrate binding and reduction are located on MOFE protein. Look at here. This is a site for Mg ATP binding present in a Fe protein, while site for substrate binding is in the Fe protein. Here the substrate and binded and reduction are located in MOFE protein, where the nitrogen fixation actually happens. Now, the reaction of nitrogen fixation is catalyzed by nitrogenase enzyme, which convert atmosphere nitrogen to usable nitrate, that is ammonia, or for the plant. In short, this enzyme is highly important in biological fixation of nitrogen, which sustain life on Earth. The transport of electron require high energy, for example 16 ATP supplied by hydrolysis of ATP, converting them into ADP to IP. So the mechanism of nitrogen fixation by enzyme nitrogenase is as follows, that is N2 plus 8 electron plus 8 H plus ions gives rise to 2 molecules of NH3 plus H2, in which this reaction is catalyzed by nitrogenase enzyme, where 16 ATPs are hydrolyzed and 16 ADP plus 16 IP are produced. So this is the structure and function of nitrogenase enzyme.